Ladies and gentlemen, please welcome Genome BC's interim president and CEO, Dr. Tony Brooks.
As you just heard, my name is Tony Brooks, and I am the interim president and CEO of Genome BC. It is an honor for me to introduce Genome BC's elder in residence, Hekwa Jila Ogwa.
Gela Kesla Nugwa Hekwa Gila Ogwa. My traditional name is Hekwa Gila Ogwa. I come from the Kwakwala speaking people, and our traditional lands are the northern tip of Vancouver Island, the adjacent mainland, and some of the little islands in between. I lived here in the lower mainland for 25 years and was adopted into the Joseph family. I'm not related to them. My last name is Joseph, but the Joseph family within the Squamish Nation. And so with that, I carry a deep gratitude, but a lot of, I want to be reciprocal. And so I want to acknowledge that we're on their unceded territories of the Squamish, Musqueam, and Tsleil-Waututh. And they've been very inviting, obviously to me, but I think all of us to be here, to live and work and play and raise our families in their territories. And I think they've been really gracious about it this whole time. Really honored to be here to do an opening for our keynote today. I'm really excited. Every time I'm involved with Genome or I do work, I'm learning things that I literally couldn't even dream of when I was a little girl growing up, and the things that are being discovered and still, you're looking into. It's just like sci-fi movies to me, coming to life in front of me. I'm going, wow, this is so amazing. I have so much admiration for everybody who does this work into connecting back to the beginning of time to today and then to 500 years from now. It's amazing to me to be able to witness that and to sit with people who have that dream and that passion to do that work. So I want to raise my hands to you today for having that passion, for having the dreams, and for doing the work. And I think it's such a gift that each of you have to. My brain doesn't work that way. And I think we all have different gifts, but to be in your presence, that you all have that gift of that passion to do this kind of work, it's really awesome to me. I'm in awe of the work that Genome BC does, and I'm really honored to be a part of this organization with so much respect. I otherwise spend my life speaking about how connected we are as human beings, and I think it's the same thing, but on a different level of what I do and what you do. I speak about our common humanity, that our diversity is important, but even more important is our common humanity, that we're all connected. We're all interconnected as human beings. And I think we forget that a lot of times because we're so focused on what success is in this country and mostly around the world, that we forget that we're all these human beings having human experiences. And I just want to bring that to the forefront of our minds, that we're all human beings having these human experiences while we're trying to succeed in the economies that we're forced to live in. And so I'm going to sing you a very short version of a love song, because for me, love is always going to be the answer. Love is always going to be the answer. That if we embrace our common humanity, that's what we're going to find. Love is in that place. And so just this quick, short version of the song. This is a love song from my nation. Gela Kesla. In my language, Gela Kesla is kind of used how the Hawaiians use aloha. It's hello, goodbye, thank you, and all of those things. But if I literally translated it, it means come well with me on this journey. So for our time here together, this is a journey in itself. Gela Kesla. Everybody come well on this journey.
Thank you, Shelley, for that beautiful introduction. Genome BC's Elder in Residence program is an important step towards advancing reconciliation while centering Indigenous voices and worldviews in all that we do. We are fortunate to gather here today on the ancestral unceded territories of the Coast Salish people, including the Musqueam, Squamish, and Tsleil-Waututh nations. We are committed to working with Indigenous peoples in ways that are culturally safe and advance Indigenous priorities as articulated by Indigenous peoples. Hello and welcome. Thank you for coming along to our annual Don Ricks Distinguished Keynote Address. We are delighted to have you here today. I'd like to extend a special welcome to the students of Moscrop, Gladstone, and Heritage Woods Secondary Schools. It's great to see you. I believe you're over here and over here, so it's great to see you, but I can't actually see you. So. It's great to see your enthusiasm for science, genomics, and of course, space. We need inquisitive minds like yours to solve problems, work together, and to shape the future of our world. This is a special year for Genome BC. We're celebrating 25 years of bringing together researchers, entrepreneurs, Indigenous partners, leaders, and policymakers to create tangible, positive impacts on our environment, our society, and our economy in BC, Canada, and the world. For 25 years, we have seen genomics improve healthcare, protect biodiversity, inform sustainability and resource management, and today we're going to learn about the value of genomics in space exploration. I'd like to just take a moment to remember the person that this event is named after. Dr. Don Ricks was a scientist, innovator, leader, and philanthropist who left a remarkable legacy on the Canadian life sciences community. He always wanted to create opportunities to fill gaps. As a physician, he saw the need to better diagnostics and co-founded a system of clinical labs across Canada to fill this need. It is known today as Life Labs. He was also a founder of Genome British Columbia. He did so with the belief in the transformative power of innovation and technology. This event is a way for us to remember Don. We work every day to follow his example by advancing innovation, seizing opportunities, and generating solutions. Through this approach, we're helping solve some big challenges here on Earth and even beyond, as Chris will tell us about. Genome BC has come a long way over the last 25 years, and I know that our achievements and all the success we see in BC's life sciences ecosystem so far reflect Don Ricks' drive and passion and knowledge. Here are some recent highlights. In cancer, more than 60% of pediatric cancer patients in Canada now receive pharmacogenetic testing to personalize their care. That's up from 10%. Sequencing relapsed tumors has led to new discoveries in lymphoma treatment, now used by the World Health Organization to establish new global standards. We've detected new viral threats to one of BC's favorite foods and largest agricultural exports, that's blueberries. And with updated tools, we're developing ways that can help our growers. Researchers, regulators, and Indigenous partners are continuing to monitor our ecosystem using environmental DNA, that's eDNA, to identify and understand changes in our biodiversity before damage can occur. In these and many other ways, genomics has stepped out of the lab and into our everyday lives. Genome BC is very fortunate to be at the forefront of this. Now I have a few housekeeping notes before we introduce our speaker today. Following the presentation, there will be a Q&A session. If you have a question, please use one of the microphones in the aisles or post your question on Slido. Feel free to post about the event on social media. The Genome BC's social media accounts are on the slides behind me. After today's talk, we invite you all to join us for a reception where we want to meet you. Genome BC will be wearing green lanyards like this. They're a friendly bunch, so feel free to go up to them and chat about absolutely anything. I'd also like to acknowledge our partner organization for this year's event, partner organizations, I should say, for this year's event. You may have already seen some of them when you came into the room. Many of you will already be familiar with Genome BC's education program, Gene School, for high school students, which has been contributing to the science curriculum since 2006. Today, they're hosting an interactive activity at their booth, and you're all welcome to participate. Just don't all go at once. Welcome to the Maya Farms team. Maya Farms is one of the latest recipients of Genome BC's I² funding, our bridge between innovation and commercialization. We hope that you get to chat to them and learn about how their mushroom mycelium ingredients are connected to space travel. We're also excited to have some people here from the H.R. Macmillan Space Center. You might have been to some of their outreach events, the planetarium, or visited the observatory. They're hosting a very special Genome BC podcast event, Nice Genes Live, at the Star Theatre on December 2nd. There's a QR code you can scan at their stand, and you can find out more about the event and get your tickets. You can see all of these stands, as I said, out in the reception area. Of course, we would like to acknowledge you. We understand the power of all this science and research, and it is even more rewarding when we get to see that you, our public, get it too. So thanks very much for being here. We also want to hear about how you feel about today's event. In the coming days, you will receive an email with a post-event survey. Outside in the reception area, there are a number of other ways you can tell us about today. Check out the board immediately to the left and tell us, left of the door, and tell us what you learned today. Or fill out a survey at the Genome BC booth and let us know what you would like to hear about in the future. And with that, I'll hand it over to Dr. Frederica De Palma, our Chief Scientific Officer, to introduce today's speaker.
Thank you, Tony, and welcome everyone. I am really delighted to introduce our keynote speaker, who is my friend and colleague, Dr. Chris Mason. And like Don Rakes, in the same spirit, Chris is obviously driven to close critical gaps in knowledge with science and innovation, as we will hear. Dr. Mason is a professor of genomics, physiology, and biophysics at Weill Cornell Medicine in New York, where he leads a laboratory at the forefront of human health, data science, and space biology. He is internationally known as one of the principal investigators of NASA's landmark twin study, in which 10 research teams examined the physiological, molecular, and cognitive effects of a year in space on astronaut Scott Kelly, compared with his identical twin Mark here on Earth. The findings continue to shape how NASA prepares for a long-duration spaceflight and human missions to Mars. The Mason labs span 3 major areas: clinical genomics, with applications in cancer, infectious diseases, and even space medicine. Beyond this lab, Dr. Mason holds appointments at the Sandra and Edward Meyer Cancer Center and the Fayl Family Brain and Mind Research Institute at Cornell. And he co-founded the Consortium for Space Genetics at Harvard. He has written several books, one of which will be available for purchase at a discounted price right outside the reception area after his talk, and the title of his book is Next 500 Years. He published more than 400 peer-reviewed papers and co-founded 4 biotechnology companies. His work has been featured everywhere, from Nature to Popular Science, and he has earned him numerous awards. So I hope that you will find Chris's science as bold, exciting, and inspiring as I do. Please join me in welcoming our 2025 Distinguished Dawn Ricks keynote speaker, Dr. Chris Mason.
Great. Well, thank you very much for the introduction, Dr. De Palma. Thank you to the organizers for Genome BC. It's a pleasure to be here. I had a wonderful chat with some of the high school students as well just before this introductory session. So, great. It's an honor to be here. It's a pleasure. I'm going to, walk you through something that sounds a little crazy. It's a 500-year plan, but trust me, we have a lot early, and then it gets a little bit more speculative towards the end. But I want to take you with me on a bit on this journey. Before I do, a quick note on conflicts. I've started and co-founded several companies and have a number of conflicts. Not as many as George Church, but I still have some conflicts of interest. But I view a lot of industry back and forth as one of the best ways to get technologies out to market. So I'm going to start with what is the vision that you've heard, I'm sure, in the popular press. You've heard this from NASA, from SpaceX, from others, that we want to eventually get to the moon and then Mars and really become a multi-planetary species, to not just go visit, but to stay there sustainably. And originally the goal was to get there by the year 2037, maybe 2035, 2039. It kept getting delayed, but this is the ambitious goal until just about last year in the fall. Elon Musk started tweeting. Now, he tweets a lot of things, and you never know what he's going to say. But in this case, he was posting a lot about how the fact that maybe the first starships could get to Mars during the next transfer window, maybe next year. Maybe we could actually have a starship go there and have uncrewed landings in 2029. Maybe have people get there, and then in 20 years, we'd have a self-sustained city on Mars, which depends on, you can see in bold, an exponentially growing flight rate of the number of rockets launching into space, which seemed a little crazy at the time because that hasn't what had been happening for the past, decades, since 1957, since Sputnik. But actually, in the past 2-3 years, this is exactly what's happened. You can see there's a number of objects launched into space, both crews and cargo, since 1957. And we now have actually this exponential increasing rate of rockets launching into space. Almost every day there is another rocket launch into space. So this projection is actually happening. So okay, maybe we could actually get to the moon and get to Mars and have this rapid pace of rockets launching into space. So it's possible, but it raises some other big questions, of course. We can now get there. You can see which countries have launched into space. But one of the big questions is, what drugs will you take if you're going to survive in space? Can the body survive a trip to Mars? There's a good article about this here. Is there a way we can customize medicine like we customize some of the treatments at Genome BC and have the right medicine for the right patient at the right dose and prepare you for this long mission? And we take this question and we put it in the context of what we've learned about the hazards of space environments. In particular, we know that the bones and the muscles deteriorate. We know that changes in their eyes. We have a lot of changes to the immune system. And I'll walk you through some of the things that we've learned in the past 5-10 years about different spaceflight risks. But we know there's a wide range of risks and hazards in space environments, and we're just beginning to understand these at the cellular and molecular level. But I want to take even a step back. When we first had this question, what happens to the body in space? The Air Force had this question in the 50s and the 60s, and they would fly, some of the very first pilots in these parabolic flights. So you get a little bit of zero gravity just for a brief burst. So you have tiny bursts of microgravity. And they would also answer some fundamental questions about science, but they would answer some of these questions, but also questions that you've always wondered. Like, for example, if you think about it, if you're trying to fly cats in microgravity, do cats always land on their feet? The answer is no. No, they do not always land on their feet. You can see here. Now, no cats were harmed in the making of this video. And I have 2 cats at home. I could watch this all day, actually. I could just watch this all day. And they're very cute, but they have the same reaction that humans have. When you get into microgravity, your body is very confused. You're trying to adapt to a completely alien environment, and you have to adapt to it. So we have been thinking about this in my laboratory for, this point, almost 20 years. I'm going to walk you through what I wrote down, actually, about in 2010. I said we should have a 10-phase, 500-year plan to understand the body and think about long-term missions to space. And this is what I wrote down first on a napkin. And then it led to a lot of work I'll walk you through. So the first phase we've already completed, and I'll walk you through some of the highlights. The first one was, as you heard, the NASA Twin City, where Senator Kelly here on the right was going to space for 1 year. Now Senator Kelly here on the left, Mark Kelly on the left, is actually, he stayed on Earth, and you can see, he's a Democrat. We could actually start to look at the genetic differences between Democrats and Republicans. Maybe, maybe we could better understand why politics are the way it is. I won't talk about that. Could you CRISPR a Democrat into a Republican? Could you modify one into the? I don't know, probably not. But we could think about these things. So, but we had the chance we went up into space for a full year, and you can see leaving there, heading up in orbit, and he posted these great photos on Insta and what was then Twitter, now X, and he did a lot of work in space. Collected hundreds of samples. Also did some juggling. This is actually very easy to do in space because it just stays there. But he did some juggling. He did some great work also growing plants and getting the experiments. He did vaccination. We got blood from space brought back down, as well as microbiome samples. I'll walk you through some of the highlights. But after a year in space, he came back, and this is actually within a few minutes of being back on Earth for the first time. He just sat down and then tried walking, and he actually, was able to maneuver. So he'd been, hadn't had gravity for a year. But then he did, even just maneuvering slightly, he was a bit unsteady because he hadn't really used his muscles that much in a year. And then after that, he had this really sharp reaction. He felt like his skin was on fire. Anything his skin touched broke into rashes, in discoloration. He had these flu-like symptoms. He wanted to go to the emergency room. He had all these really stressful responses. And he, really had this harsh and sharp reaction. And our big question was, of course, was just why? What had happened inside of his body to elicit this response? So what we performed was a longitudinal multi-omics, meaning many layers of biology analysis of a year in space. In particular, the quick highlights, because we've published some of this a few years ago, was we wanted to see changes in molecules that went up or down at the same time in concert, which often indicates co-regulation. So in particular, we look for things that would increase in space, like proteins, small metabolites. We could see genes change their expression. But one thing that was really surprising was telomeres, actually, at the end of your chromosomes that keep your DNA intact. They got longer in space, which was the opposite of what we expected. We thought the stress on the body, the changes, we'd have shorter telomeres, meaning he would almost look older. But in this case, he looks younger because his telomeres got longer. So this was the opposite of what we expected. And that made us think, well, he was eating well. He was actually working out almost every day. He also got a little taller because he gained about 2 inches. So then some headlines came out that he got taller, and he got younger. He lost a little bit of weight. You get taller, younger, and you lose weight. It's the best diet plan ever, is just go to space. But it didn't last forever. It went away when he came back to Earth. But it raised this question of, well, one, was this real? And how do we put it in the context of other people who have stress? So then some people had reached out who were climbing Mount Everest. They said, oh, Dr. Mason, I'm about to climb Mount Everest, and I have a twin who's staying at sea level. Could we do a study? And whenever someone offers me blood and stool, I say yes, basically, is generally my answer. So we set up an additional study to say, well, we should take a look. And indeed, people who climb Mount Everest also got longer telomeres, you can see quickly on the right. And then we saw this in 11 other astronauts that we tested as well from bank samples. So, okay, it seems to be consistent. But we want to also not just have a PCR-based method. We want to get a way to do sequencing. This is a genome center, after all, so I want to talk about sequencing. So we use this what was then new at the time, the Prometheon, and is basically get out the new instrument and developed a new method, which we call edge case, you can see here. But basically, you want to take all the molecules of DNA, and when you have your chromosomes, you want to layer them down almost like you're building almost like layers of a dip or something, and then want each one to map onto the genome and figure out exactly how long the telomeres are. So we did this with these nanopore reads. We could actually stack them up and then do a better alignment, and we could see indeed, actually in space in the middle, his telomeres got longer on average, but we also saw some shorter telomeres, so it was a spreading of the distribution. But we confirmed that was real, so okay, that seems real. But what else changed when he was in space? What's driving some of these changes? So then we looked at the gene expression, which is how genes get turned on and turned off. Figure when you go into your kitchen, you turn on light switches, you turn them off. The same thing happens in your genes, you're turning them on and off. And we saw all these colors are many, many genes changing their dynamics. This was the immune system, genes for DNA repair, hypercapnia. Well, that's hypercapnia. That's when you have too much carbon dioxide. So what does that mean? How could that be happening? And even Scott Kelly wrote a whole book about his year in space. He complained a bit about this. So we checked the flight logs, checked the space station logs. There's oxygen, carbon dioxide. We're pretty stable. But then we talked to some other astronauts and asked them, well, what's it like in space? Why are we getting this carbon dioxide signature? And it turns out when you breathe in space, it's not the same as on Earth, of course. So you can see you get these mini clouds of carbon dioxide that can probably form in front of your face, which you think is what we're seeing in the blood, where you can see exemplified by Jack Fisher here, which is kind of funny way to see how air moves in space. So if they don't, astronauts have complained if they're not by all the fans or if they don't move enough, they will get these headaches. So we think that explains some of the hypercapnia. But I want to go back to the rash. That was the thing we really wanted to think about, what happened there. And this is where we looked at the immune system in particular. The day that he landed back on Earth we saw these large spikes of hundreds or thousands of % higher than baseline, which were for the things like C-reactive protein, IL-6. These are molecules that really give large inflammation in your body. So we could see these here and realized, oh, this is probably what was driving that really sharp immune reaction in his body. But then we thought, well, that was Scott Kelly, but how does he compare to other astronauts? So we took data from NASA and said, okay, we have about 30 other astronauts. And this is what's, is a distribution of what other people look like for these 6-month missions versus a year in space. And you can see pre-flight, in the middle of the flight, post-flight. You can see the blue line is Scott. So he sometimes was the highest number ever observed for IL-1RA, for example. Never been seen before that high. But other cases, he was flatline and had no real changes, cool as a cucumber. So what this indicates to us is that everyone is different when you go to space. There's stress response, there are changes in flight. Just like on Earth when you take a medication. You want the right drug for the right person at the right dose, at the right time. And stress in the body, we all respond differently to stress. So we realized at the beginning of kind of precision medicine for astronauts, because we can see there's some pretty clear differences between the groups. And we said, overall, it's good news. They mostly recovered. We looked at many layers of biology, these different omics layers. But that was kind of the end of 2019, 2020. We thought, okay, we need, that's the end of phase 1 is what we thought would be new gene engineering systems and astronaut studies. But what I'd written, 10 years prior actually came to pass, which is, okay, we need to do more astronaut studies. We need to start looking a bit more into the future. And right around that time, SpaceX had just started saying, okay, we want to actually start to fly civilian crews into space. So we had a unique opportunity to work with SpaceX and the medical operations team to say, okay, let's plan out some of the same experiments. Let's do the same profiles. We look at blood, stool, urine, skin swabs. You can see here the stool is brown, the urine is yellow. We just couldn't resist. And the blood is red as well, which is, it's evocative, I think. So we kept those colors. We had a long debate in lab meeting about, should we make the poop brown? And we kept it brown. Anyway, yes, we kept it there. But we want to study everything we could before, during, and after spaceflight to see what changes. What's responding the most to spaceflight with this protocol? So I had a chance to go to the launch, and you can see here when the Falcon's being loaded up with fuel. You can see it's launching. This is right at sunset. It made its way up into the upper atmosphere. Heading up farther and farther north, and actually went up to a higher altitude than the space station or the Hubble. You can see here in this kind of top line orbit, about 590 kilometers at apogee. And when I got up there, I got this beautiful view of the planet as it was starting to make an orbit. But these were 4 people who only, basically 6 months prior didn't know they were all going to be astronauts. Well, one guy did because he was planning the mission, but the other 3 on the right had no idea. So they've been trained, prepped, and flown in space all within about 6 months. They get up there, they take a selfie because they're excited they're in space, but then they all stick some other selfies. They're up there for the Inspiration 4 crew. But then they got to work, actually did blood samples, did microbiome swabs, checked to see if we could get this new Butterfly IQ device, which can do ultrasound in space, for example, getting this to work. So they did hundreds of blood draw samples, all this work, collected all the samples. We were all very anxious, but then they came back down from their orbit. This is right before they splashed down, made they were okay. We were all very anxious, but then, they were cheering, excited, because they weren't dead. Basically there's that moment of terror when you're coming down as a rocket through the atmosphere. And then we had some members of the lab get up and get ready to get the HEPA filters and the swabs, everything out of the spacecraft when it got on the boat. And what we just presented last summer was a big summary of this other mission, which was the Inspiration 4 crew, to see what happened to their body and see if we could confirm some of the previous study findings and then expand on this a bit. So this was a paper we just published last year. And what's exciting about this, again, this is a crew that, if you remember in 2021, there was a Super Bowl announcement of who wants to go to space. You could apply. And so this is a crew who had never been trained. We did in-flight, real-time training for this Butterfly device. So you can basically take your jugular vein or look at your eye to do an ultrasound, a kind of a real portable diagnostic device you can see there. And also the Apple Watch we flew for the first time. That also worked okay. It worked fine, actually. And you can see their bladder and jugular vein, they did okay, but the eyeball was a bit harder. But they managed to actually get trained and then run this protocol. So that was good to see. But then we also want to see what else happens at each individual cellular level. So in this case, we were able to do what's called the 10x Genomics multi-omic kits, where you can look at expert genes going up and down, as well as chromatin, so is DNA being open or closed through this protocol. And we looked at each individual cell type. And it turns out the monocyte cells were the most impacted by spaceflight. But we also saw a lot of T-cell activation signatures. You can see here, probably can't see in the back there, but just each cell type had a unique signature in how it responded to spaceflight, which was interesting to see. And we hadn't been able to tease that out before. And the other thing is we looked at, these are some of the more differentiated cells, but you see the stem cells on top. We wanted to see, do we see evidence of other stem cells? So we actually looked at cell-free RNA, which is something, if you take your blood and extract all the DNA or RNA, both of them have signature. In this case, the cell-free RNA showed us which tissues were contributing. You can see platelets, monocytes, again, these different cell types are part of each of the colors. But one that really stuck out was the stem cells. So right after the flight and then decreasing afterward, the signature of stem cells seemed to spike up. So that was another indicator of stem cell changes during spaceflight, which had not been seen before. So okay, so these stem cells are changing, their bodies are adapting. What about telomeres? Here in gray, we did confirm their telomeres also got longer in this very short mission. So we could see in short missions, long missions, it got longer, but not as much as the year-long space flight. So we think it is a kind of a dose response. The longer you're in space, the bigger the change, which makes some sense from what we've seen for the body. And so we could confirm some of this here. And you might be thinking, okay, what's happening? Is it the radiation? Is it the stress in the body? Why are telomeres getting longer? It seems so peculiar. So thinking about this, what happens at the end of your genome, you have your subtelomeric region and the telomere, which again is that cap at the end of your chromosomes. And we know some of what happens normally, but we want to look for telomerase, which is an enzyme that extends it, and that was actually pretty stable. So that wasn't it. We looked at, you could see radiation seemed to be driving the elongation as well, but then we looked for tera, which is this non-coding RNA. It's a piece of RNA that helps guide this elongation. You can see that black arrow. And we said, okay, if that's what's guiding it, we should see more of this RNA molecule in these missions. And indeed, this is one of the things we looked at for a specific paper. In red, you can see in flight during the twin study, and in the day after they landed in the Inspiration 4 crew, we see this increase of this tera RNA. So we think that's what's driving this telomere response, and we can see it with radiation and also again across these missions. So that helps explain it. But then we wanted to look at other questions. Okay, if any of you had, a bad roommate, you've been in a room for a while. Have any of you in this room ever had a smelly roommate? Anybody? Probably somebody. I think everyone, you have. You're just not raising your hand. I know you have. But it's questions of microbiome come up. So is the microbiome changing how you respond? And we had this question for the twin study. We looked at this a bit, but we had a unique chance here because we wanted to see, did their skin microbiomes converge? If you've never seen this paper, it's a great paper by Jessica Green that looked at after you play a sport like roller derby, for example, if you swab the skin before the game and then again after, the two teams are genetically more similar. They have rubbed their microbiomes onto each other. And this is basically, just because they're bumping, they're grinding, they're playing at baseball, this could happen, but more so like basketball. There's any sport where you're having any physical contact, you will exchange your microbiome. If you go into a crowded elevator, you could have a little bit of a microbiome exchange. In fact, I recommend next time you're in a crowded elevator, just say out loud, it's time for microbiome exchange. And then just see what people's reactions are. I've found people are very confused, but they're all intrigued. And they're all like, yeah, I guess we are. And they don't want to think about it. Every time you shake hands. Anyway, this happens all the time. It's just that now we have a way to quantify it, measure it, and see it. So in particular, we looked at, this is the largest microbiome study that had been on 758 samples across 10 parts of the body to see what had changed. And so we're going to look at what changed within an individual or between them and also between the spacecraft. In particular, we made this plot that shows the proportion of changes. So the thickness of each line shows how many species were moving between an individual or within or between different crew members, and also in red is the space capsule. So the walls of the space station carry their own microbiome, and those also would get onto your body. So we can see this constant exchange. You might be looking at the armpit and think, what was going on with two people's armpits? Because it was this massive transfer, an explosion of microbiome diversity. So you might be in the shower, tomorrow and think, oh, I'm just in the shower. I'm just washing my armpit. It's like a sad, dark, dank place of despair and dirtiness. But it's not. Actually, what you should do is you should look at your armpit and think it is an extraordinary utopia of microbiome diversity and new discovery and new species. It's excitement every time you look at your armpit. Don't look at your armpit like it's a dark place. It's an exciting place which can sometimes transfer to your friends. That's all I'm just saying. So that happened during the mission as well. So there was a whole range of other studies. We published actually 44 papers across many teams to look at these data. So this was published last summer. Those are some of the quick highlights. And we also launched a new data portal in this biobank where you can see here, we're storing this for other people who wanted to examine these data. Which then helps ask the question, okay, what's next? What are we doing over the next 5-10 years? We are basically using this model of the Space Medicine Biobank, like what's been seen for the UK Biobank, or the United States has this program called the All of Us, where 1 million people are getting their genome sequenced. And we're trying to link that to medical records. So these are some of the models that we're also deploying. Actually, one of my genome is up in the All of Us program. You can see here, hi, Christopher. This is my DNA profile and my risk of disease, so I can log in and see things. And it'll ask me questions like, will I like cilantro as one of the things I could look up on my little portal. And I love cilantro. I could fill a bathtub full of cilantro. I could fall asleep in a bathtub full of cilantro. I love cilantro. But for some reason, the database thinks that I have a high chance of disliking cilantro, which makes me very sad because I love cilantro. But the important thing about this is it's not perfect yet. You have to think about when you build a model for something as complex as do you like cilantro or how tall will you be, it might not be perfect today, but the ability to have access to the data and build these better models is what will drive the field forward. So I'm okay knowing the database is completely wrong today. I just want to log in again in 5 years from now and see that it has been updated, which they're working on that now, which is great. So along these lines, one of the projects we're also starting is the Million Phenomes Project, which is not just genomes, but also epigenomes, proteomes, all these other layers of biology like we've done for the astronauts, which is also going to connect all these different stool samples, blood samples, many other layers of biology. The other thing we're doing is we just completed the Polaris Dawn mission, which was to also collect many layers of biology here. And getting portable blood processing methods, here like an Easy Draw or Tasso devices. One place we just used them also last fall, and we'll be doing again in a month, is during the New York City Marathon. If any of you are in New York City, in about a month, there'll be a marathon. And since the route goes right by lab in Manhattan, we had an idea in lab, like we should probably draw blood from people that are running by our lab. Because again, if someone says I have either blood or stool or urine, I'm always like, yes. That's basically my answer. So we thought, well, this is a unique question because people often ask, is going to space like running a marathon? I was like, well, now we could actually find the answer. So we did this protocol here was Tasso and also some Wattman blood collections. This is me in the middle of collections trying to flex so I didn't look so gross, but I was covered in sweat there. And everyone in the lab kept offering me beer, and I was like, I don't want beer. I'm in the middle of running a marathon. But we did a collection. Unfortunately, Mar was the one that got tasked with bringing this back to lab. You can see his face there. He was, and he took that back. But anyway, so the point of this is to get more of these portable methods actually deployed. And the other way we're doing this is getting this so that you can have the sequencer work in space and do sequencing wherever you may be, whether it's space or a remote area on Earth. And we got the chance to do some of this for flight validation, but also the first chance we got to try this was also on a parabolic flight with Andy Feinberg. He had a chance to fly it, so we thought, let's see if we get this working essentially on a parabolic flight. And this was on the airplane. And let's see here. So if any of you have done pipetting in lab, you know that you want the tips to stay stable. Ideally, you have a stable platform. But in microgravity, you can see here, things do not stay in place. Andy's trying to stay focused. There's people puking behind him over there. He's trying to keep the pipetting going, a little bit of microgravity, because you get to the bottom of the parabola, eventually you'll lose the microgravity, but fell down eventually. But he was a great sport. He managed to load the flow cell. We showed that it was possible to sequence in zero gravity for the first time. Showed that this was kind of a proof of principle, and published this as well, that nanopores are ready for liftoff. And what's very exciting is then the next astronaut going over to the space station was Dr. Kate Rubins, who was literally a trained virologist and a sequencing expert. So it couldn't have been a more perfect choice to go up into space. And so we've been working a lot with Kate more recently to have these protocols work for anybody, anywhere. And that includes any new space stations. That's what I want to also plant the seed for all of you in the audience, especially any of you all in high schools and younger scientists, is what's coming in the future. These portable medical technologies are helpful on Earth, but also there will likely be several space stations, possibly 4 in the next 5 or 6 years. One of them is Axiom Station. So in this case, you can see this is the rendering of what it'll look like once it's completed, possibly by 2030. Orbital Reef should be done by about 2031. That's what it's rendering. So it's also called from Blue Origin. And Orbital Reef kind of sounds like a coral reef in space, very evocative. VAST is going to probably fly their first module next year, hopefully, the very small one. And they'll also try artificial gravity on their station. So this is the third one, and extraordinary to see that this is also coming. And eventually there'll also be, probably by 2032, is Starlab, also with Voyager Space, which it looks like this from the top, and will actually be about this large. It's just 3 dedicated decks, a science deck, a central greenhouse. And these are being planned. So in the next 5 or 6 years, there may be 4 whole new space stations. The ISS will be deorbited by 2030, maybe 31. So this is an entirely new realm of space and research that's coming up above our heads. And so what that means, of course, and if you're thinking about microbiome transfer, there will be more of these kinds of transfers. And then it starts to raise the other question, if there's other transfers, if we start going into space, to the moon and back, could we get transfers between the space station and bring it back to Earth? Could it be dangerous? Could it be a mutated strain? We've seen many mutated strains in space. And to really examine this question, we really need a way to monitor any molecule that's on this planet or space or the moon or Mars. Really, we need a system that could examine a planetary genomics. We want to look at any molecule that's ever been seen on our planet and know where it's been and how it got there. So, which means a global microbiome. And so I got interested in the microbiome as a geneticist because I always want to see what DNA, when I sequence something, where it came from. But I also got interested in microbiome research broadly when I became a father. So if any of you are parents in the audience, you know what happens. Like you've got a cute kid, you're going around New York City, you go into the subway. And at one point, I don't have a picture of it, but my daughter was grabbing the subway pole and she straight on licked the subway pole. And I had a moment of pure parental terror because I thought, she's exchanged microbes with her mouth and the pole and the pole in the mouth, and something has happened there, and I just wanted to know what it was. And I'm a scientist, so I go look online, I look in the literature. There was no real studies. There had been one study in Portugal on a bus, one by Norman Pace that looked at the air a little bit, but nothing about what's on the surface. We have no idea what was there. I said, how is that possible? So I thought, what we need is a microbiome map of the metropolis, or what the New Yorker called the metropolome, is what we should really have. So we started swabbing everything. I thought about dropping my daughter off at daycare. I was like, wait, I'm dropping her off at daycare before I go to lab. I could swab her mouth before I drop her off. And then at the end of the day, I come back and I swab her mouth again. And I can see like what are the microbes she gave to all the other kids. And then I thought, but wait, then I'll be a creepy dad, like swabbing all the kids at daycare. I'd probably need an IRB. This would be complicated. That's going to be too weird. I said, what if instead we just go out and start swabbing everything? Let's just swab everybody, everything, everywhere. Let's just go and swab. So I had a bunch of summer students in 2013, and they said, well, Dr. Mason, I'm interested in epigenetics and inherited disease and complex human disease and all these questions. And I said, yeah, I hear you. But that's not going anywhere. We've got your whole life to study that. But it's never been done before. We've never had a genetic map of a whole city. So I had these undergrads, and I said, listen, how do you feel about wandering New York City all summer and systematically swabbing every station in triplicate? And they said, yeah, let's do it. So it was great. We built an app, a crowdfunding campaign, and a whole protocol. And then went out and got all 468 subway stations in triplicate, went out and swabbed them. You can see here in the map. Brought them all back to the lab, extracted 1,500 samples, sequenced them all, took all these fragments, about 10 billion fragments of DNA that we sequenced, compared it to every known species to see what we could find. And then we actually found here is, you can see, the pie graph in blue is unknown organisms. It didn't match any known species. We'd never seen them before. Now you might think, is the New York City subway full of aliens? And it feels that way sometimes, but really it's because the databases were and still are incomplete. We're still finding entirely new species quite literally under our fingertips in the subway. You can see it red. We found a lot of bacteria, we found viruses, we built density maps of everything that had been found that was a known species, but half of the world under our fingertips is still unknown in the subway. It's something that's populated by millions of people every day. And so this was extraordinary to think about, but then it raised other questions like, well, what's there? How does this impact human health? How do we describe this to the public? How do we compare this to other species? And I started getting a lot of people sending me things. Like, for example, things that would just see, like, for example, this is a guy who said, hey, what's going on with this guy? This is down near Coney Island, near Steelwell Avenue Station. This guy just swabs with his finger and then eats everything that's on the... And I don't have a protocol for this. I don't know. And that, and some people post online like, immunity, that's you're going to build an immunity one way. So I started getting things like this. And then people started saying, okay, you've published this. And the number one question I would still get for a while was, okay, Dr. Mason, I just want to know, is it okay to lick the subway pole after your daughter? And I would, of course, say a very complicated response. So, well, it depends on, the immune system of the licker. It depends on how long it's been there, the temperature, the humidity. There's a lot of factors. It's probably fine generally. Most species are not harmful that we found, but it really depends on all the factors. And I then learned a lesson I'd like to teach everyone is that media is not the place for nuance, because this is the headline that appeared. So that's technically true. It's probably fine, but this is what. So if you talk to a reporter, they'll quote you down to 2 words. Just gotta remember that. That's not so bad, because I did say probably fine. But the worst thing is, give them media, give them pictures, because if you don't provide a picture, sometimes the reporter will say, well, I need something for this copy, I need a picture to go with the story. And then someone in the office is like, I don't know, here, just use this picture. I took this on the subway, and this is what goes with the story, which, is the most disgusting thing I've ever seen, basically. So, and my name is now forever right below there. You can find it forever, it's right there. But my point being, be careful when you talk to the press, and there's a lot left to discover. But I bring this up because there was an intriguing dance with the media. But what's interesting is I've been pleasantly haunted by that study ever since we finished, is who's there and what are they making? This blue part of the pie graph, these completely unknown species, literally under our fingertips, in the soil outside, in the air, and the subway poles. Who are they? What are they making? What molecules are they making? So this led to, again, a lot of people sent me funny stories in media, but then also people would say, what does Paris look like? What does Hong Kong look like? I saw your study. So one of the best things about science is if you just put your crazy idea out there and publish it and say, well, here's what we found, a lot of people come find you and say, well, what does it look like in our city? And so this actually led to the founding of a nonprofit and a big global consortium we're calling Metagenomics of Subways and Urban Biomes, also looking at design. And we thought, let's build out a global map of what's in all these cities, because we just don't know what's there. And we can then also track antibiotic resistance. We can mine the data, look for new biomolecules. And we thought, okay, well, let's build this consortium together. And if we could, we would swab the whole planet at once. But of course, that would require a big swab. So instead, we set up a coordinated fashion. So once everybody wake up and pass the international date line, we begin swabbing. Every summer, it's called Global City Sampling Day, where we coordinate with all the other teams around the world. We begin swabbing. These are a collection of a lot of the cities that have contributed every year to swab their city with the same protocol, same swabs, same dual matrix tubes, essentially collecting everything and then building up the metadata to see what is in the urban environments of the world all around us. Is mostly really of discovery to see what's there. And you can see, again, some of these maps, all of it's on the website. And this has led to some people who got creative on Twitter, X, for example, Naranjan in Singapore got really excited with his team to get creative in the lab. Alina, she actually was detained by some of the security officers and then had to be put in the cell. But then when she got let out, she said, wait, before I leave, though, I'd like to swab the cell. And she did. That was dedication to science. So she's still one of our favorite stories from the consortium. So we published some of the first work on this in 2021, which led to a threefold increase in the size of the tree of life. We found 11,000 new viruses, several hundred new bacteria, literally on the subway poles. Note the date there. That's in the spring of 2021 when we announced 11,000 new viruses, which was in the middle of COVID. But mostly phages. They were not many new dangerous viruses. Again, most of them are pretty inert towards human health. And excitingly, we also didn't just find new biology, we also found a lot of new potential enzymes that could be used for CRISPR and new CRISPR arrays, which represent the history of what bacteria have been fighting. So we can see this continual movement of microbes around the planet, especially with what happens with air travel and people moving around the planet. So we have this new way to quantify the movement of molecules around our planet. And we did eventually create that planetary genomics map. We made this global KMER index. You can go to this website as part of the paper. And if you would type in any genetic sequence, it'll pop up where has it been seen before in any database and what has it been associated with. So this is a way you can we have a first draft of a planetary genomic profile that we're continually updating with Andre Callas and Gunnar Reich, who are phenomenal scientists at ETH Zurich. So we have this beginning of, I'd say, planetary genomics. But that's kind of the first half of the talk is what we've done and more recently and looking ahead. But I want to switch gears to looking a bit more forward for the last part. And then I'm going to end on a bit of a philosophical note and end in a strange place at the end. But I want to think about what I again wrote on that napkin is like in the phase 3, we should think about long-term trials for genome engineering. We should start to look at off-target effects. We should think about, could we build armor for the genome, for example? Because if you look at what was in the Scott Kelly, you can see this is the amount of radiation in the back. It might be hard to see, but this is the amount of radiation for a Mars trip for 3 years in red versus Scott Kelly's year-long mission, 146 millisieverts versus the Inspiration 4 mission, 4.7 millisieverts versus, say, X-rays, or if you were a witness to the Hiroshima bomb, an awful atomic explosion. See what are these ranges of radiation you could be exposed to? Mars would be beyond all of them, right? So it would be very hard on the body. So it raises some interesting questions like, well, could you, of course, if you can physically protect astronauts, we can pharmacologically protect astronauts, but could we genetically protect astronauts? Could we turn on genes to help them survive before they get there? There's an article about this from Antonio Regato. But then should we do such a thing? What are the ethics of making someone fit for another planet, which I want to focus on for the last part of the lecture today? And how do we do such a thing? And it seems crazy to just even ask the question at first. Like, how could you even ask this question? Until you think about where else do we use genome modifications for therapeutic purposes. In particular, we do this for CAR T therapies, where we modify T cells, take them out of the body, reinfuse them back into your body after they've been completely reprogrammed to fight cancer. And we and others have published on this. There are now hundreds and hundreds of clinical trials modifying immune cells and putting them back in your body that are completely genetically reprogrammed, but for therapy. And so this is not completely out of the question. It certainly has been happening. But what about other ideas? Like, could you, for example, take genes from the tardigrade, this cute little water bear that can survive in the vacuum of space? When its genome was first sequenced in 2016, the researchers noticed it had this protein that made it so that DNA could withstand radiation. It had about a 40% reduction in DNA damage. And we thought, okay, well, that's extraordinary. You can use this tardigrade protein and quickly put it into a human cell, and you get this benefit. So you basically take the evolutionary lessons from one species and then you apply them to a human cell. So we've built a cell line in the lab that actually has constitutively active, meaning it's always present, this protein from a tardigrade in the human cells. You can see here the D subprotein labeled here in these little green speckles. And where we see double-strand breaks after radiation, we can see the tardigrade protein working in the human cells. And we have actually seen about an 80% reduction in DNA damage using this protein, inside of a human cell. And that's using genome modification, which maybe is a bit more, I think, severe. But you could also, if you're thinking about what's in the cells and the DNA you have, you think about the fabric of life, think of these threads of DNA and the genetic code. What if instead of changing the threads, you just change how they get used? And this is basically CRISPR systems, but instead of gene modification, you do an epigenetic modification of this. So these are what's called dCas9 systems. So as a quick technical background, the Cas9 you've heard of is the enzyme that helps CRISPR function, but you can have a deactivated Cas9 or dCas9. So it still has the homing beacon function of CRISPR, goes to one spot, but doesn't cut. It just brings with it a different protein that turns either histones, to change epigenetics of histones, or change DNA methylation, like DNMT, for example. So you can go in, and so imagine if you went to your kitchen, you want to turn on a light switch or turn it off. You're just turning things on and turning them off. But imagine if you could do that in a human cell. And this is something I fantasized about when I was like a high school student, if you will. They're like, oh, what if someday we could control genetics down to one little spot and change them? It seemed like a complete science fiction, but today it's working in our lab as we speak. It's using these kind of methods. So, and the reason I bring this up is, if you can do epigenetic modification, of course that's important, I mentioned for spaceflight, but also we know that in cancers, for example, acute myeloid leukemia, a lot of the consistent mutated genes are epigenetic modifiers. So, and to get a bit technical of what happens inside the cells, these genes that control DNA methylation, if they're mutated, for example, genes like IDH, IDH1 and 2, they no longer, you don't have this continual cycling of methone hydroxymethylcytosine. So these molecules that normally are cycling through various states in your cells, they get stuck. And when that happens, you have this buildup of epigenetic changes. And we and others have published that if you have these mutations, you can have different paths for leukemia. You can have genetic or epigenetic evolution that can happen. And so, we've looked at this change of how the leukemias evolve. But what we end up doing often for these kind of cancer studies, we can say, what's happened? You've been mutated, or there's an epigenetic change. What happened, and what does this mean for your prognosis? And if you looked at cancer therapies, you want to be on the black line. You want to know that you're a low-risk patient, that you're likely to live, for thousands of days. Not for hundreds of days, right? You want to be on that black line. And so we often do this in a lot of clinical care, is we will get a better way to stratify high risk and low risk patients. And ever since I started looking a lot in cancer genetics, I find it very dissatisfying to just make this plot. You're like, okay, I can tell you whether you're bad or very bad. And if you're a patient, you don't want to know how bad you are. You want to know, what can you do for me, right? You want to, what can you change? And so for epigenetic differences, historically, we've only been able to do this. But recently, because of these dCas9 systems, and we're starting to get them to work in cells, we can actually go in, for example, it could be for cancer patients or astronauts, we can go into these specific loci and turn on genes. You can see here 3 examples on the left from lab, or turn them down, sometimes 90, 95% turn down specific genes. So what I'm very excited about is looking ahead, we have this technology to go in and at very specific loci, turn things on or turn them off. Which again, I fantasized about when I was like in high school or in early undergrad. It's like, what if that could happen someday? And it's already a reality, which I think is just extraordinary to see this possibility. So if that's possible, then you start to get a bit more creative. You can start going a little bit more in the future. It starts to get a little bit more crazy, but come with me. It's going to be interesting. Could we test these new protected genomes in space environments? Could we do this in phase 4? And if we're doing that, what would we give these genomes? If you're going to a faraway planet, you have very few resources. You don't have everything you want normally with you. So you have to basically, could you think of other ways to get self-reliant? So for example, how do you get your vitamin C today? There's a pseudogene in your genome that used to make vitamin C, but no longer does. If you want to make vitamin C, some people get it from drinking mojitos, for example. That's one way to get orange juice. You could get that one way to get your vitamin C. You don't want to get scurvy. But what's interesting about vitamin C is I think all of you know you need to get vitamin C because you can no longer make it in your body. But if you look historically, In bold are species that still make their own vitamin C. Dogs and cats, for example, do not get scurvy. They can make their own vitamin C. Most dry-nosed primates, like humans, for example, have all lost the ability to make vitamin C. We used to have it. Other mammals have it. Other animals have it. But we no longer have it. But the gene is still there. And so you could, for example, what if we just bring it back? What if we start to make our own vitamin C again? That's one thing that might be helpful for long missions. And I think, okay, that's one idea. But also, we have 9 essential amino acids. You need to eat these in your diet. Otherwise, also, again, you get very sick and eventually you would die. But why is that? Why do we have to go forage around in the forest to get amino acids? Don't we know what the genetic circuits are to make all the amino acids? Indeed, we do. So here, if you modify the existing genetic code of human cells with a little bit from yeast and a little bit from a couple bacteria, we could complete these circuits and then go deep into space and make all of your own amino acids, which I think would be nice, and also, again, self-reliant. Or you could even go a little bit deeper into the future. And like, if you know you're at a risk for, say, a disease gene like hypertrophic cardiomyopathy, could you modify it as an embryo? You could even do it right as an embryo stage. And again, this seems a bit unusual, but then if you think about other examples where we've taken a gene that was once working as an embryo or a fetus and bring it back, we know it can solve problems. So for example, one that was discovered in the past few years is when all of you today are using alpha and beta hemoglobin to carry oxygen in your body. So you can see here, when you're a fetus or an embryo, and it's different. But when after you're born, generally, these are the 2 main kinds of hemoglobin. But when you were a fetus, all of you had a different kind of hemoglobin that was working and carrying oxygen. And the gene is still inside of you. It's just been turned off. So what if to treat, say, sickle cell disease, you just turn it back on? And this is exactly what was tried in a clinical trial in 2021. First tried to say, let's just turn back on fetal hemoglobin. Will that work? You can see it's here in blue. And also for beta thalassemia. And then it worked so well, 2.5 years later, it's an FDA-approved therapy. So that simple concept of, can we turn back on a gene that's still hiding in your genome to cure a disease has already been FDA-approved. And so one of the patients from his really extraordinary story, if you haven't read it, but Jim Alleghieri, actually, he had sickle cell, had never been able to work out at all, and then he climbed Mount Kilimanjaro, and he was actually at NextMed, a conference that was earlier this March. So a really phenomenal story. And then more recently, other cures, like what if right at birth we can see CPS1 mutations, for example, could you use CRISPR to do cures right at birth? And here too, it's no longer hypothetical. This has been done and published also about a month ago. So this raises interesting questions of, if we know which genes we want to keep an eye on or maybe target, could you have precision, protective alleles for long-term spaceflight? And this is kind of a summary of what's published a bit in my book, and also more recent work in the field of all the genes that could help bone density or muscle strength or ways you could have a healthy immune system, things that you could think about. Now, we have not tried this yet. I was once introduced by someone at NASA that said, here's Chris Mason, he's genetically modifying astronauts and sending them into space. We have not done that yet. We're still thinking about it. And then everyone in the audience tried to find me and wanted to be mutated, but that was not what we were working on. We want to think about this. We tried this first in cells, then we tried in mice, then we tried maybe in primates. We're still far away from clinical applications, but we do have human data and human cell lines for some of these genes of target already, which is just fascinating to start to think in the next, again, 25 years ago, it was science fiction to go in and modify gene expression, but now we can do it easily. And so I think what this gives you, or gives me this view, this hope for, is that you have a new kind of liberty. We all think of liberty, life, liberty, the pursuit of happiness in the US, but we'd like to think about, what if you had cellular liberty? Like from the very first cell, you can do anything you want with your cells. They're your cells, you should be able to do what you want. So for example, what if you want to modify your cells to have them have GFP and walk into your own shower and have it glow green with your own cells? I don't know why you want to do that. But you can if you want to. You should, in theory, should have this ability. So if you can do that with your own cells, could you do other things? Like could you, haven't you always wanted chlorophyll? You look at the plants, you're like, that's not fair. They just get to sit there and get all their food from the sun. I would like to have green skin. What would that take? What if I want to go down by the pool and I want to lay my skin out? I want to get all full up on the sun's rays. Am I the only one that's thought of this? You've all thought of it. I know you have. You look at the plants, you're like, I want chlorophyll. I would like to be and lay my skin out. So we did the math on this. Like how much green skin would you need? And if you lay it outside for 1 hour, what's the degree of epidermis expansion you'd need? It's about, you need to go, for 1 hour, it's about 2 tennis courts worth of human skin is what you'd need to just lay out. And then it creates a new kind of social environment. Like, would your friends play tennis on your skin? Probably. Would it tickle? Probably. It's interesting social interactions, but this is interesting about like if you had that, and it's based on the fact that you could make one cell could become any other kind of cell. So for example, What if you what if you have a problem with your egg or sperm, but you want to make your skin cells into egg or sperm? That's also been done in mice. What if you want to have 2 mom or 2 dad children? Also been done in mice. It's possible to have one cell become any other kind of cell, which means you have total cellular liberty. You can do anything you want with any of your cells. And this, again, at the first blush might seem odd, but we know that there's 3 parent children, for example. You can have a mother and a father, but also a mitochondrial donor. You can have 3 parents. And again, if you have cellular liberty, could you have tissue liberty? Like what if you get uterine cancer, but you want to have children? You don't have a uterus anymore. Could you have an exogenous womb that gives you all the capability to develop and essentially, and this has been done actually for lamb, you can see her in this kind of creepy video, but you can see her entirely exogenous exowomb that's present. So again, the kind of a creepy looking video, but shows it's possible. And I go on at great length about exowombs. Actually, you can see again, it's in this paper. I talk about it a lot in the book that's in my first book. And at one point, my mom asked me, because she read the book, she said, "Chris, you go on for a long time about these exowombs. And I couldn't help but after a while take a little bit of an offense because you talked so lovingly about these other uteruses." And I said, "No, Mom, I really had a great time in your uterus." And so for the record, I want to say here publicly, I had a great time. It was a fabulous home. I was born 3 months early though, so I did have to leave early. But one time my mom came with me to a talk in Boston and she sat in the front row and I told that story again. And then everyone in the audience started clapping in applause for her uterus, which was a weird situation. But I, again, am very grateful and I had a great time there anyway. It was not like this. It was much better from what I remember, which I don't remember any of it, of course. But in any case, anyway, so it's this idea that you can have cellular liberty from the very first cell. Like you're not beholden to the genetic shuffle you got as an embryo. You could have liberty in any cell, but then this starts to get creepy, right? Or even, is it unethical? Like, can you really select the best embryos? This idea of embryo selection has been discussed, of course, and critiqued well by others. And the idea is relatively straightforward. If you think you have 20 embryos in front of you, for like in vitro fertilization, you can combine to make what's called the polygenic risk score and say, okay, we just want to take all the risk genes and the high risk and low risk and put them together. And ideally you have a high risk in red or a low risk embryo and just, pick the best one. It seems pretty simple, right? But then the challenge is our knowledge of genetics is still being teased out in human genetics. So in this case, imperfect measures give you imperfect metrics. So actually it turns out if you try to do this, for example, what's your risk of schizophrenia? You mostly just predict the ancestry of the person more so than you do their actual risk of a disease. So we're still teasing out these polygenic risk scores. And I propose that we shouldn't just say, are you at a higher or lower risk for the disease, but you have to put a genetic technology into the context of what is the cost of it? What does it look like across multiple generations? Which is socioeconomic impact, what the cultural impact, what are all the different burdens that it might place on society? So I think we shouldn't just think about, whether you should pick an embryo a certain way, but what is the other factors that come into play, which are many. And again, it seems abstract, but you have to think, could you do embryo selection or even change a species? Some of this we know can be done. If you've never heard of the silver fox domestication experiment, it's a fascinating experiment where you put a bunch of Russian geneticists in Siberia. What could go wrong? And they're all out there and far away from the rest of the world. And they had a simple question. Could you take silver foxes and make them into dogs? Just take the docile ones, have them breed, but take the aggressive ones, just let them die of old age. And could you do this? And so this was an experiment started back in the 30s and 40s in Russia. Within a few generations, they started to get juvelinized facial characteristics. They got floppy ears. You can see they got like a shorter snout, getting like these different characteristics that made them a bit like dogs. And their brains changed too. One of the students in our lab, Lenore Pipes, characterized their brain tissue after they died to see even their brains were changing. And so this means you could, within a matter of a few generations, have a fairly different gesundheit, a fairly different species present, possibly. And so if you think of people on a different planet, it would happen probably much faster. And so this, again, it seems abstract until you think we know some of this has been done before. So the other thing to think about looking further ahead is if we get to the near 2100, we could start to look at planets that are farther away. So for example, could you go to an alien ocean? Could you find creatures that can survive at the bottom of the ocean? We know some of this also happens on Earth. There's creatures that can survive. This is a postdoc in the lab, Braden Tierney, who's now started a nonprofit called the Two Frontiers Project. Basically finding cyanobacteria that survive right by these carbon dioxide vents right by the volcano. And you can see here they can actually eat carbon dioxide. So you can actually find these extremophiles that can help you eat up carbon, for example, at the bottom of a volcano, which has been extraordinary. And so eventually we'd want to use these same ones to maybe treat climate change on Earth, but also could help you survive on Mars because you want to basically turn carbon dioxide there into oxygen. And so the goal would be long-term to make it so Mars would be like this, self-reliant, makes all its own medicine, captures most of its waste. This will take, of course, many generations to get to this point, to go from tens to thousands or tens of thousands of people. But this is something that's possible. Again, I'll be dead long before this happens, but building these structures that many people will benefit from, I think, is a great value and honor to do so. So that would be looking further ahead. And then we're getting out towards the outer rings of the solar system. So we think, well, could you survive on other places like Titan or these other farther away planets? And this is where we take lessons from even more extreme organisms. There's a project called the Extreme Microbiome Project, where we've been sequencing different microbes all around the planet that have survived in really strange environments, including, even in some cases, the space station. And there's one like Delanina salina, which is basically a polyextremophile. It can survive in high radiation, high salt. It actually can be, eventually just multiple stressors. It survives fine, and so we use those as lessons for what could survive someday on Mars or maybe planets that are farther out. And then if you get to that point, it starts to raise these other questions of planetary protection. If we can make an organism that can survive on almost any planet or selected planets, could you then send them to Mars to have plants grow there? Could you seed plants and other organisms to start to grow on these planets that are far away? When should you do such a thing? Should you do such a thing? Which again seems abstract, but we know that NASA in this looking far ahead is looking at, and we have one proposal that's been active with them to take plants that could grow in lunar regolith, or taking model data from essentially deserts, for example. So NASA's already actively thinking about ways to engineer soil so that plants can grow on the moon or someday other planets. So it's not abstract, it's ongoing. And the exciting thing is, it's not just the planets near us. We could eventually think about having a 3-planet flyby mission, which we've been planning since last year, is to go past Mars and then Venus, then back again. If you want to see more about this mission, you can this work with Sarah Seeger is the Morningstar mission, hopefully coming next year. We can actually send a probe to Venus. It's relatively cheap to go to Venus these days. It's only a few $100 million, not that bad. It used to be much more, but you could actually get there fairly straightforward. And so that opens up, once you start to go to Mars and Venus and other planets, I want to propose to you, then you can have a new also kind of liberty. You could have planetary liberty. Could you go live on any planet that you want? So today, you can't live on any other planet except this one. We've been perfectly situated to Earth for many, many millions of years. But what if you could go to another planet? Could you go to Mars and come back? What if you engineer someone to only live on Mars? I would find that to be a reduction of planetary liberty. The goal would be you could go to live on any planet you want to, much like today. You have continental liberty. You can go anywhere you want on Earth and go check it out and explore. But the ability to go to any planet is something we don't have yet, but we could have. And then if we have it within this solar system, it starts to raise questions in the later phases. Could we start to go outside our solar system? Could we go to a new exoplanet, which is a planet in another solar system, and maybe get there? This is a list of all the exoplanets found since the early 90s. We used to know of 0 exoplanets, of all of human knowledge, but now we have thousands of exoplanets. Each color is how they were found. But we have there's several hundred of them that are in the habitable zone, meaning they likely have liquid water. Some of them look like this. So we have many planets that you could go to today, maybe pop into the solar system, maybe land. It might be a little hot, a little cold, but it's possible that they're within a few tens or dozens of light years away. And you could go there and maybe survive today. And they're actually many of them Earth-sized. And so this creates something called an Earth Similarity Index. For all these possibly habitable planets that are not that far away, how similar are they to us, and could you go there and land? And so this is something called an ESI, which is kind of a complicated metric, but basically the closer you are to 1, the better it is. The more it is like Earth. Earth is 1 because it's our own reference. But you can see this, like Mars and the Moon are not even that good. They're below 0.8. They're only good because they're close. The other planets that are basically above 0.9 or 0.8, which are blue or purple planets, are not that far away and would be much better for us to get to. We could probably even survive on them, or maybe survive on them today, certainly much more than Mars, which has almost no atmosphere and is very cold and is a lower gravity. So these other planets are not that far away. And if you actually take the list of every known exoplanet that's been found that's either purple or blue, again, it's possibly good candidates for us to go to. This is our galaxy from the top and from the side. And this is every known exoplanet that's been discovered in white, or is it possibly habitable? You can see these blue and purple spots. There's a lot of them. And we've really just started looking in the past 20, 25 years. So there's probably a lot more that we'll be able to find. So it means that there are all these places we could possibly go. And so in the later phase, it means that you could look at phase 9, where could we, by the end of the next 500 years, have all the technology to get people into a starcraft, have them go there, have multiple generations, live and die in the same spacecraft on their way towards these planets, which we've already found that you could potentially land on and get there? And the answer is somewhat yes, actually. We already, even just today, and we'll know more and find more very soon, but we have these planets already But then, you might think, that seems far away and you'd be stuck on a spacecraft, but you could have all the Netflix you want, all of Amazon Prime, but it'd be an unusual environment. You'd be stuck in one spacecraft with many generations living and dying in one space. But then it's really just a question of scale. This planet is one large spacecraft, and all of our generations live and die on this spacecraft. Now, there's a lot more to do. The weather's better. You got a lot more space, of course, but the concept is the same. It's just bigger. In this case, if you could have someone in a spacecraft, you'd be on your way towards the biggest adventure of all time, and there might be many of them. But this is the point in discussions with people, at bars or restaurants, I get a number of questions. People say, okay, well, this is all very fascinating, Chris, but I don't know, why should we do this? Why? Well, I get a number of questions. The first question I get is, Chris, what's wrong with you? And then I go, well, how much time do you have? And I've got there's a lot of things. I was born 3 months early. I left my mom's uterus too early. There's a lot of things you could imagine. But why? The biggest question is why do this at all when we have poverty, there's disease, there's social, economic problems, there's strife, there's wars? Why spend time, energy, money, blood, treasure on this versus anything else? And I want to give you a really solid answer as to why, and then end on a bit of a philosophical note. So first is, we can make progress on both. It's a false dichotomy to say you can only do cancer work or you can only do space work. Clearly we can do it in my own lab, but also as a society, we know we can make progress in civil rights legislation, for example, in the United States, and land someone on the moon at the same time. It's a false dichotomy to do only one or the other. The second reason is one of an intangible asset, is hope. It's something that when you look at the sky at night and you see all the stars, you imagine them as a place you can go to, not run from, that the future is brighter than the past. In particular for my own daughter, who's now older and runs into the ocean at Venice Beach with her friends, I can go towards the skate park at Venice Beach, and we have a discussion about what would it look like on the moon where it's 1/6 gravity, or Mars where it's 38% gravity. And that conversation is not hypothetical. I'm having an actual conversation about missions being planned to go to those planets that someday she may skateboard on the moon. And it's a real conversation. And that is an extraordinary future that we can have and talk about together. The third reason is one that maybe is the most important reason. And to close on is that we know life, for example, in North America, a quarter of the birds have been lost over the past few decades. We know that our modern lifestyle is probably destroying our microbiome diversity. We're losing species before we can even find them. And this is not the first time. If you look at the history of life on Earth, this is over the last 500 million years. What's the temperature been on our planet? We've had a world without polar ice caps in orange. We've had a world with polar ice caps, you can see here in blue, and it's gone up and down many times. You can see this is the history of the temperature and life on Earth, and every time the temperature has changed too quickly, that's when the mass extinctions have occurred, 5 so far. Of course, we're trying to avoid a 6th mass extinction right now if the temperature increases too fast. But even this, I want to take a step back and think of like there was a time when there was a snowball Earth. Step back, that 500 million year chart, and take this in the context of the history and likely future of our planet, and we're basically right about there. And this very nice but very temporary temperature canyon, at some point, the sun will get bigger, and essentially the oceans will boil in about 1 billion years. And when I was a kid, I always thought we had about 4.5 billion years before the sun gets engulfs the planet. So we've got a little bit of time. But when I was finishing updates on this chart, I realized we've only got about 1 billion years. And I was moping around the house, and my wife then said to me, why are you so sad? What's wrong? And I said, I just thought we had more time. And she laughed because I was still 1 billion years. I said, well, but I thought if you thought you're going to live to be 100 years old and someone said you're going to be dead at 20, you'd be devastated. And I feel the same way about this chart. And then that assumes everything goes slow. There's no asteroid. There's estimates that the next supercontinent may doom mammals in a far future extinction. Our planet is estimated to look like this in about 250 million years, which would be pretty hot. And so then it means that everything is at risk maybe sooner than 1 billion years. And then if you think about who's at risk, this is something I think about all the time is, of course, there's producers, consumers, decomposers. This is all the tree of life we think of. There's 3 kinds of life. But what I want to argue to you and have you remember is that there's a 4th kind of life that's unique to this, which is the only kind of life that's aware of this sort of frailty of existence and extinction. Only the 4th kind of organisms is really us. It's something that can guard against this, the guardians. Quite literally, we are the guardians of the galaxy, is what I'm telling you, because only we are aware of extinction. Only humans, to our knowledge in the whole universe, have this awareness at all. That gives us a duty towards all life present and future, and maybe even the past. If you think about what is coming soon and has been starting to occur, is discussions with George Church a few years ago is like, what if we could bring back the woolly mammoth, which was at first very much a harebrained, crazy idea, but now has actually already been completed. Earlier this year, we brought back the dire wolf, ourselves and many others at Colossal Biosciences. And it was a fascinating moment because we thought, if you want to bring back a creature, what do you pick? How do you know what you're going to bring back, and what traits are there, and how do we bring a map of genes to specific phenotypes? And it's already the first de-extinct animal has just been born earlier this year, and we can use technology to protect and revive other species, and eventually get to other solar systems. Use this technology, and then you might say, okay, fine, Chris, or the guardians of life on the universe. Okay, smarty pants, fine. If you go to the next star, eventually that star is going to get big and run out of fuel. The next star is going to run out of fuel. This just seems ad infinitum. What do you do, smarty pants, when is the universe going to end in a big crunch or a big rip? What do you do then? What are you going to do to just keep running from star to star to star? This just keeps on and on. What happens then? And I think what's interesting is in the last week, actually, the most recent estimate is that we might only have 19.2 billion years left before the big crunch happens. Based on last week's estimates. So it's probably going to be a big crunch. And if that's happening, then we really have two questions. Should we prevent the implosion of the universe or allow self-destruction of the universe in the hopes that life will arise again in the next universe? And I think this is the biggest and the last ethical question we have to face. And I think faced with these two options, I think we do what we've always done. We survive. We keep life alive because we don't know if life will ever come up again, if this is the only universe it'll ever happen. So maybe we have to carve out a section of the universe that survives the big crunch, or maybe restructure the universe in the ultimate sort of engineering challenge. Should we do this? Can we do this? I think it's what we have always done and I think need to do. And that's kind of the thesis of what is a duty towards life itself in this universe and any universe. You can, of course, there's some copies outside. It's recently been translated into a few other languages. And I want to close also with thanking everyone in the lab who makes this possible. I could not stand out here without having an extraordinary and amazing team that I'm honored to work with in the lab. This is an older group of photo. This is a more recent group photo of everyone who, again, is extraordinary to work with. Of course, funding from the NIH World Quant collaboration with NASA SpaceX for a lot of the missions. Gratitude to many people in the lab. And finally, the biggest thanks goes out to the patients, their crews, and their families, and all the data, resources, and samples they donate. And I'm happy to take some questions. Thank you for your time. I promise not to share any more weird subway photos. That's right.
Thank you, Chris. This was wonderful. Thank you very much. Okay, so we can begin our question and answers now. So I'll just, you have 2 microphones either side of the aisles. You can also use a Slido. So the information to connect on Slido is on the screen, I hope. And we're also on social media. So if you post anything, feel free to use the hashtag. Okay, so do I see people? Yeah, okay.
Very much. A couple of concerns in the introduction. The video of the cats, physiologically, they might not have been strained, but psychologically, I have to check their expressions. The other comment about rockets, exponential use of them, makes me think about how we once thought of the oceans as being so vast. And, pollution, could that be harmful for our thin skin of atmosphere? But my real question has to do with the first part where it's concluding about the astronaut and the twin, the study of the other astronauts too, that after a year's time, overall it could be okay. But it hasn't been measured and it's unknown if astronaut being 5, 10, 20 years, whether it would be truly toxic or lethal. And I thought about capturing all the identifying the viruses. Could 3 times factor be still almost such a staggeringly small number of the total population of viruses?
Good questions. I'll start in the order. So cats, in theory, they're okay, but we don't know their psychology. Fortunately, we don't use cats anymore for experiments. Really, the Air Force doesn't either. That was some of the very, even Laika was the first dog that was sent into space. The Russians sent Laika first because they were too afraid to send a person, and so the unfortunate dog had to go. So humans are the best model for humans. And at some point you have to test in humans, but there's always a balance between if there's something that seems really dangerous, they often would, of course, do mice and a lot of animal studies. But these days, one good thing is you can use organoids or sort of avatars. You don't have to use even full animals. You can get a lot of information from collections of cells, which don't have a lot of more of the ethical problems. Second question on the rocket fuels. This has come up before. It's like if you're burning all this carbon and all this waste going up into space, what's the sustainability argument for sending giant rockets in space? And yes, it's an exponential rate, but it's at an exponential exhaust rate and pollution rate. So relative to the scale of the atmosphere, it's not that big yet. But if it went 100 or 1,000 times more than that, it would start to have an impact. And I think that is something we have to keep an eye on. But it is also in the grander scheme, it's not as large of a factor for the atmosphere. But I think it is an important question because it recognizes that we have an ability to do geoengineering, planetary scale engineering, which the best example was when we realized the problem with the ozone layer, basically in the 1980s. As a global community, we got together and said, oh, that's bad. I don't want to just walk outside and get sunburned. So how about we get together and stop CFC production? Now, what's interesting, I think most of the people in the audience, you probably think, oh, the ozone hole, we solved it, right? But if you actually look, the hole is still there. It won't be gone until about probably the 2060s. So if you look at projections, it's getting better, but it's not gone yet. So I think the challenge with geoengineering and planetary scale engineering is that small tweaks can have decades-long impacts. And so we have to have, as a global society, a capacity to have multi-generational planning and responsibility, which has not yet quite made the mainstream, but I think the discussions like this help people think about that. And geoengineering, we should quantify and measure it every time we do that. And then for the last question, long-term follow-ups for the astronauts, most of them are really healthy, so they've generally, the only risk of cancer seems to be rare cancers, and really we're too underpowered to make any big claims. I sometimes joke it was the lowest powered study that's ever been done is one person, so we can't really say, even when we looked at a few dozen astronauts, we can't really say too much that's prescriptive yet, but as we collect more and more samples and more crews over decades, we'll be able to look at long-term risks. But so far, there don't seem to be too many long-term risks because the astronauts generally live pretty long and healthy lives.
Hi.
Yeah, sorry.
My name is Elena Lopez. I'm just a clinical geneticist. Kudos to your mom.
My mom's amazing. She's the best.
I've housed two of my own. What do we know about human reproduction in space?
Great question. So there's actually a company called Spaceborne United, which if you want to check out their website, their goal is to try and get pregnancy in space, which raises all sorts of thorny ethical questions, like how can you really have informed consent for the couple? And certainly you can't have it for the embryo. But we know that, let's start with some mammals. We know that mice have gone up, and actually I should say have gone up pregnant and given birth, and some mice have come back, embryos that were fertilized, and they have had problems. So we know that there is, at least from mice data, there is a substantial risk of doing full embryogenesis in space. There's a chance that maybe it's not as bad as we think, and there were just some differences, but humans could be different. We mostly don't know, and this is going to be one of those kind of experiments where you could probably never get ethical approval. It'll happen probably on accident, because if we start doing these really long missions, if you get people in a small room together, historically, maybe there's a little vodka. Maybe there's a little loneliness. I mean, something will probably happen. I think my guess is that we may not find out through an actual clinical trial of embryogenesis in space, but just people will get lonely and bored or excited, or I'm not sure what's going to happen, but I think we will find out accidentally. But we think development can definitely occur. It's just a question of how much risk there is. And so we'll also, that's another case. We're probably not going to fly full embryos, but you can fly embryoid bodies, which have a lot of the same totipotent cells, and study some of these on upcoming missions. But it's a great question we know very little about. We know basically nothing for humans. Technically, officially, there's never been human sex in space, according to officially. However, there was a couple that got married, two astronauts. They didn't tell NASA that they were married until they got to the space station. And so there's a presumption that maybe something happened, but we don't. Officially, it's never happened, for the record.
Hi, thank you for the really interesting talk, and I commend you on your very diverse work. I have a lot of questions, but for one question, I see it is already kind of posted on Slido now, but as someone who has no personal interest in, living on Mars or going there, I really liked your kind of point on the kind of simultaneous progress, and, we can still kind of work on things here and this kind of future space goals as well. So I would like to, you're talking about the altitude and marathons and kind of getting data from that as well and comparing to some of this astronaut stuff. How do you see all of this kind of work that you're doing with the astronauts in space kind of applying to us here and now, and how could you see that like benefiting and kind of getting, two things out of that?
Yes, how do you do both? And actually, I did come up with one of the questions here is, how do we help? There's often a push that NASA has to. How do we do work in space but have it benefit Earth? Or they often will say their tagline is, research in space to benefit Earth. I actually take a slightly different view from NASA in this. I think it's okay if there's something we're doing that only benefits people in space. It's great when it does both, but there's so much we don't know about space, including like embryogenesis. And if we really are going to start becoming a spacefaring species, there's so much we don't know. I think it's okay if sometimes it really solves a space-specific problem that helps people live or reclaim waste or process nitrogen. But the good thing about a lot of those, the NASA's economic impact has been expected to be in the future maybe a 9-to-1 ratio. There's a study from 10 years ago that said it's almost a 10-to-1 ratio. For every dollar spent, what does the economy get back from what NASA did because of patents, because of startups, because of Velcro, because of GPS, because of miniaturization of what you need? Space is a great forcing function of, okay, I want to do a diagnostic or I want to do anything I can, but it has to use almost no power, take up no space, and be very easy to use so that someone who's never been trained can just do it. I mean, that's basically what we all do for become portable COVID diagnosticians during the pandemic. Like, all right, I'm going to stick this thing up my nose. And it had to be so easy that anyone could use it. And I did several FDA tests during the pandemic. They have to test it on old people, young people, blind people, anyone who's never seen a test, and someone who's really an expert. You have to make sure everyone can use it, because the FDA, at least in the US, they want to make sure that it's, that it's at least 95% of people can take something they've never used and actually get it to work. So spaceflight is a great forcing function to make sure whatever we develop there, like portable sequencing, for example, is a great example where we can sequence anywhere, and that helps with diagnostics, disease surveillance. So I think it's, as a geneticist, I want to sequence everything, everywhere, all the time, of course. But that's a great example that helped us. That miniaturization helped us do more field work. And I think any of the miniaturization technologies for fuel waste reclamation, water purification, 97% of the water on the space station gets reused. Like, imagine if we did that on Earth, right? And so some of it's just, we don't do it on Earth because it's just, you just go get more water. But if we get it even cheaper and even better, then you could have your house reclaim all your water. Now, there's an old joke that Scott Kelly was going around, and people would say, is it true that when you were drinking coffee that was, it was urine from the day before? And he'd say, well, it wasn't just my urine, because basically it's everyone's fluids, right? But once you purify it, it's good water, right? So what if we did that, where there's dirty water anywhere on Earth? So I think there'll be a lot more that'll come from that kind of work. Yeah.
Thank you.
Hi, thank you very much for the excellent talk. Lots of topics, very interesting, very entertaining.
Thank you.
I wanted to circle back to the earlier content on the telomeres. So you showed some data that it was being mediated largely by the tera-RNA.
Yes. Yeah, the non-coding RNA.
Did you also look at what was actually triggering this response? Like, was it being triggered by radiation exposure, or was it carbon dioxide elevated levels? And then follow up to that, is this like a beneficial response? I mean, we always see that telomere length is associated with aging, so we think of it as beneficial, but so many biological responses are very context dependent.
Yep.
So in the context of space travel, is this a beneficial thing?
So good question. I'll start with the last one first, because like longer telomeres, also cancer cells have elongated telomeres and can divide almost in perpetuity. So it's not always good to just have constant long telomeres. And so we don't know, I'd say. We don't have enough samples to say is it good or bad. And even for most of those dynamics, we think things are changing. I don't know which of them are adaptive versus maladaptive. I think they're all clearly changing. But for example, we took that one paper and said, what if we looked at every drug that could target those cytokines? But would you want to decrease all those cytokines? Because maybe some of them are helping the inflammation response, but they might be functional. So the short answer is we don't know, because we don't have enough data or interventions yet we've tried. And then for the mechanism, you can actually take cells and just irradiate them, and that is enough to actually elongate the telomeres as well. So we think the driver is likely radiation, more so than CO2 or just fluid shifts, but from what we've seen. So yeah, good question.
Cool. Thank you.
Thank you.
And then
I was thinking we could answer perhaps the most voted here on Slido.
Oh, it's voted. All right. Yeah, the number.
Can you speak of the mechanism behind the microbe?
The radiodurans surviving. Oh yeah, so it's not everyone knows. NASA's been taking these trays and letting them sit outside the space station, being irradiated and exposed to the vacuum of space, then brought back in. So radiodurans, we're actually trying some of the purified protective proteins from radiodurans in human cells, actually this week in lab. We think some of it is the desiccation pathways. I don't know if I can see who's anonymous there, but anonymous number 1, likely desiccation-related pathways, and also some of their radioprotection proteins is my guess. But I'd be curious to see for the strains that are more tolerant of spaceflight than others to look at the genetic differences. But we mostly don't know yet. I'm guessing what I just answered, but we mostly don't know. So that would be a good PhD thesis, actually, right there.
Chris, also the other anonymous question, that question you asked for.
What is the probability that life exists on these exoplanets, given that nucleic acids and peptides form so easily? It's a great question because, indeed, most asteroid studies have shown that almost every amino acid has been found in asteroids. Almost every nucleotide that exists, every nucleoside, has been found in asteroids and comets. So the presumption now is that the raw ingredients for life seem to be pretty ubiquitous. The presumption is that there's billions and billions of stars, and every one of them probably has at least several planets around them. So there's billions and billions, probably trillions of planets. And if that's true, what's the probability that life exists? So the real answer is we don't know, and so far it's just us, and that's why I think it should impart upon us a sense of duty towards life. But if I had to guess and be pinned down, I'd say there's probably bacterial life somewhere in the universe, but I don't think any low probability of complex life. The main reason being it's 13.8 billion years that the universe has been here. We needed a few billion years just for the universe to cool and for elements to form, then supernova to make heavier elements. So we're already 4 or 5 billion years in. Then the universe had to start to glom together to actually get organisms. Life existed on this planet for probably 3 billion years in a very simple form before it got complicated. So I think that probably means other planets, if we're any guide, also need several billion years before they'd make complex life. So because of all that, my guess is there's bacteria out there somewhere tooling around, but that complex life might just need more time. I think in the history of the universe, we're maybe the first ones at the party of complex life, and we should go out and help others if we can. That's my view.
Hello again. I don't want to overstay my question, welcome, but since the mic was open, I thought I had one more question I wanted to ask about your urban microbiome study, which is really interesting. I know you mentioned students sampling it, in triplicates over one summer, but how much kind of longitudinal data have you looked at, or how stable is an urban microbiome, or do you know, or what do you think in terms? I know there's a lot of species that you found, but what are your thoughts on that?
Yes, well, we see even at one station from the first paper we did. There's even within hour by hour, we'll see a lot of movement of species. If you can just basically, we swab the exact same kiosk where you buy tickets every hour on the hour. Although not every, so I had a student who was supposed to start at 9 a.m., but it didn't get there until 9:15. So the figure goes 9:15, 10, 11, 12. Anyways, we almost got it every hour on the hour, but it was up for the whole day. And there were a lot of changes during the day, but if you think about it, it's thousands of people touching the surface. So we weren't surprised that there were a lot of changes, but we just wanted to see what it was. And then to the first question, we have 10 years of sampling data now, so we're in the process of doing all the extractions. And we're going to sequence them all and have the data available hopefully within about a year of 10 years of longitudinal sampling across about 60 cities around the world. Mostly because my daughter licked a subway pole and I just wanted to know it was there. So it hasn't stopped since then. I just need to know what's out there. So, but what's amazing is how many other parents are out there who are like, I also have seen licking or other problems in subway. So there's so many of the collaborators are, people who are just curious. It's been a fun family event. Actually, there's a group, like during COVID, we worked together. When Ukraine was invaded, some scientists left and came to the consortium. So scientific consortia, if you do them great, become like little families, actually. So it's been a wonderful community. Let's see.
Have a designer baby ethics question.
How do you tackle design? And this came up in the Q&A with the high school students. So kudos to the high school students for having a question of this, almost the same question. So the key 3 words are unmet medical need. And if you're addressing an unmet medical need, that gives you a good reason and also ethical basis for any intervention. If any of you know He Jiankui, the Chinese scientist who made the CRISPR babies, he deleted what's called CCR5 so that the embryos couldn't get HIV. But outside of the other technical problems with his consent form and the IRB, some other issues that he did it so they weren't really well informed, which is its own problem. The medical problem was that there was no need to do that therapy, and also that therapy it was an earlier version of CRISPR, which I'm sure was not as good in terms of having off-target effects. So, but nothing he did, there's other ways to avoid HIV during childbirth. The parents had HIV. There's also other ways to, even if you get HIV, to have antiretroviral therapy and live a full life. And so, in this case, the designer babies, if you're going to design a baby, like edit an embryo, you need to have an unmet medical need that you're addressing. Otherwise, you're just doing exploratory, dangerous research. But the bigger challenge is with IVF, you're not designing the baby, you just have choices. So Orchid is one company I talked about that does this, there's others. And there, as a matter of course, for IVF, you generally have at least 4 or 6, maybe a few dozen embryos, and it's very easy to test them all. So there, it's really a designer or baby selection ethics. And there, the polygenic risk scores are also being offered. They have problems already. I think it'll be there, you're within your parental rights to say, I want to pick the embryo that's the least likely to get disease. It's not irrational, but the problem is that the algorithms are very early and imperfect in many cases. So I think I would prefer us to wait before we did that for say another 5 or 10 years, but the service is already active in the United States. So I'm wishing for something that doesn't happen, that's already happening. So I think the key phrase then would just be, can we have truly unmet medical needs that are being addressed? Can we empower people to be as informed as possible? And can we have them be for not cosmetic features like, hair color, eye color, but have them be really for disease avoidance, which I think is more grounded in ethics. That's it. And then, okay, and then genome.
It's a similar question, but it's...
So good question. So if you can't see in the back, genome engineered therapeutics are only approved for serious illness, which makes sense. How do you get them through regulation for healthy individuals? Like if you just want to go into the mountains and activate some hemoglobin for the weekend and say, I'm going to just get a little CRISPR game on for the weekend, and I just want to, turn these on for a little while. Now that didn't make sense when there was only CRISPR as an option. Like who's going to mutate themselves for the weekend? That doesn't make sense. Although I have some friends who, when they drink enough, I feel like they're mutating themselves for the weekend. I have good friends from high school who've definitely been mutated for a long time. In any case, but if you can do epigenetic CRISPR, you're just slightly turning things on or off. That actually gives you an ability to say this is for, say, a stressful event. Like I want to go, like I mentioned the Mount Kilimanjaro climber, that was for an actual CRISPR therapy to modify his genome, but you could imagine, what if you're at low hemoglobin and you're like, well, I'd like to go, I'm healthy, I'm mostly healthy, but I want to go on this big event and I'm going for my honeymoon to climb mountains, I'd like to just do a little tweak there. So I think it'll start with edge cases like that where people have you don't need to go climb the mountains for your honeymoon, but it'd be nice to. And so I think you'll see things like that where also, you first have to do safety, efficacy trials, clinical trials, and long-term follow-up to make sure if you do that it's really safe. But that's happening now. So I bet you in 5 years from now, we'll start to see recreational epigenetic, well, maybe 10 years from now, but not that far. I think we'll see it relatively soon.
You could do EPA like Lance Armstrong.
Yeah, or you can do like Lance Armstrong and do EPA. That's right. You can do. That's right.
Over there, there's a question.
Yeah, you could. Yeah, there are other easy ways to do some of these treatments as well. Yes. Yes, in the back.
Thank you for a very informative speech.
Thank you.
My question is going towards the ethics part a little bit, and specifically 2 parts. What kind of safeguards can we, as a research community, put in place to prevent political and religious interference in the type of research, the type of methods, the type of results that we're looking for? And how can we then, part 2, make sure that those safeguards are actually effective?
Yeah, great question. So the safeguard. So and you mentioned by politics and religion and like society, all of which have different levers or parameters. I take a few quick examples. Like one, for example, xenotransplantation is where you can actually modify pig kidneys or pig hearts. And if you're on the wait list for a transplant, I think as many of you know, there's not enough organs for the people who need transplants. So what's been being developed by eGenesis, United Therapeutics, and other companies is let's slightly modify the pig genome so it can grow the organs and harvest the organ and then give it to someone who needs it. Now, there's a bunch of questions there, like if a pig gave you its kidney and it died, should you have the bacon from the pig? I don't know, maybe. But then other questions, religiously, if you're Jewish, should you take a xenotransplanted kidney? Can you? So most rabbis that have given small edicts on this have said that because it's medically necessary, if you're going to die or take this pig kidney, that it's okay to take it. But that was a religious question of, well, is it or is it not? Would you, or how are you going to enable this? So you need the religious leadership to say, we're blessing this. This is, if the choice is, the value of life and dying versus taking something that is of porcine origin or a pig, which normally you wouldn't do as a Jew, they've blessed it or said this would be allowable. And so that was one example. And I think for other cases, a lot of these other therapies, you have to engage them early and often and say, ask the question about this technology has let us make these kinds of cells or create these modified organs. what do you think? What would you like? Even simple question. Actually, in one part of the book, I mentioned if you're on Mars and you're Muslim and you want to pray towards Mecca, you know, Mecca would just be, you wouldn't know where it is. Its Earth is like far away. So should we make a second Mecca, like so that every planet gets its own Mecca, so then you'd know which way to pray as a Muslim? The translator for my book said, well, Chris, I don't know if I'm going to translate that part of the book because I might get murdered or you might get murdered. And I said, okay, it's up to you if you want to. I said, I think it's a question that I think doesn't warrant my assassination, but is just a good, it's just a question. I think if you can say, we don't know what the rules are for going to another planet because we've never done it before, I think it's okay to even have a bit of humanity, sort of just to go easy on ourselves and say, we've never had a ability to ask these questions because we've never gone to another planet and lived there. So it's okay to say, for everyone just to say, we don't know what the right answer is because we've never done this. So no more questions.
If you would permit me just a bit of a follow-up for that, the safeguards that we would need to put in place to prevent interference in the research so we can continue to do this research and find out what the answers to the questions you've just raised.
I think the safeguards, you mean to prevent, because you don't want to stop either the implementation or the research. I think to keep the research going, you would just start small. I guess the best thing is just you have a question, do it on a few cells in one lab or two labs, sort of very small. And the safeguards in society are, I guess, engagement and really before we, for example, the CRISPR babies, but he did it in secret and then like announced to the world, aha, I CRISPR'd embryos, which is the worst possible way to do, there were no safeguards. He just, there technically were some at the school, but not really. So I think often and consistent engagement is better than just jumping ahead. So
thank you.
Yeah, thank you. I think
Chris, before we end, can you tell everyone what happened to astronaut Kelly's telomeres when he came back?
Yes, we have. They have stayed. They have not gotten longer again. So we have monitored them. They've stayed. We also do follow up for what's called clonal hematopoiesis and other mutations. So he has technically been more mutated, but so has everyone else in this audience over time, actually. So, but we're seeing, is it different from what you'd see from other people? And so far, no. So far, he is the normal amount of mutated older man, basically, as all of us are, I'd say.
Thank you very much, Chris.
Thank you very much. Thank you.
Thank you, and thank you everyone for the great questions. And I would like to invite Genome BC's Chair of the Board, Dr. Kausar Samli, to come to the stage, please.
What an incredibly fascinating narrative journey you took us on. Thank you. So we learned about armpits. We learned about toe jam. We learned about Chris's fascination, or lack thereof, of subway stations and poles. And I'm compelled to think of your closing remarks, particularly around progress, duty, and hope. To me, these things embody a kind of devotion of the human condition, a kind of love. That takes me back to Elder Shelley Joseph's opening remarks about love that is the commonality of humanity. Thank you so much. As a gesture of our appreciation and for sharing your insights today, we would like to present to you a talking stick. And this talking stick is hand-carved by Wesley Weiss, a Coast Salish and Musqueam artist. You can see a picture of it up. Please try not to lick this. The talking stick is used in many indigenous cultures as a powerful communication tool. It provides a unique opportunity to share perspective, wisdom, and experiences, particularly for the person who holds the talking stick, as they are granted the floor. This ancient tradition embodies the principles of equality, ensuring that every voice is heard and respected in the communal circle. If you'll join me on the stage, In gifting this talking stick to you, Chris, we honor your presence with us tonight and all that you have shared with us, the insights that you have discovered and surfaced, and the contributions that you are continuing to make to the life sciences community. Thank you.
Thank you. Amazing. Beautiful. Wow. Amazing. Yeah, I really, this is beautiful. Thank you very much. I didn't have other planned remarks other than to say, again, really honored to be here. This is, the questions were great. I really encourage everyone to keep debating and thinking these questions during the cheese and the snacks and the drinks. You know, I really mean it that we, you know, love is really a through line for a lot of the work, and it's a love of genetics, of curiosity of the cells, of what's so much left to discover. And I really think that students and really everyone here, you know, we have this duty towards continuing the discovery, but to do it in a responsible and ethical way that brings everyone together. And so I think I'm a humanist, and I'm such a humanist, I think we could make it through the end of the universe. So I encourage you all to think about everything we do between now and then. And I'm a humanist, and I love it because I can't imagine a universe where there's not poetry and talking sticks and music and art and science and people having weird conversations and people telling stories about licking subway poles, like all this comedy and even the small moments of humanity, I find endlessly wonderful and things I would like to preserve, you know, for this universe and anyone that comes after. And so, yeah, thank you so much. Thank you. That's amazing. It's amazing. Oh yes, I get this one. Middle, yes. Yes. It's beautiful. Thank you. Thank you very much.
So before we wrap up the formal part of the night, I'd just like to acknowledge a few more people. Dr. Federica De Palma for helping shape this year's event and facilitating the Q&A session. And a special acknowledgement to Sally Greenwood and her communications and societal engagement team, as well as our volunteers, for bringing together all the little details to make events like this possible. And not only tonight, all the other events and outreach activities that we do throughout the year. So join me in a round of applause for those. Thank you. So we've concluded the keynote part of the event. Please join us at the reception where we can continue these discussions. Remember, the Genome BC staff will be wearing their green lanyards. Our partner stands are outside. Please visit them. I'd just like to remind you our next event is a live recording of our Nice Genes podcast at the Star Theatre in the planetarium on Monday, December the 2nd. And the episode will be about the genomics of music. Finally, if you've enjoyed our speaker today, Chris's books, his signed books are outside. And looking to the future, it's only cash only. It's been wonderful having you all here tonight. Thank you for coming. We love this opportunity to meet with you and get excited and interact about science. And we just love that you're so passionate about it. So thank you very much and good night.